Hey guys, sorry I had to end my last video a bit early because I had a remote appointment to attend. So this last video goes along with chapter 15 and is all about immunoassays, specifically ELISAs. ELISA, E-L-I-S-A, is an acronym that stands for Enzyme-Linked Immunosorbent Assay. And it's a very common assay that's done in research and also in diagnosis. And what is important to me for you to understand about it is that so many of these tests, these lab tests and assays, are really developed to sort of mimic what happens in the body. We're basically just, like PCR, for example, is just it's DNA replication that happens in cells, except we've just taken the key components and stuck it in a tube and used it to our advantage. So much biotechnology is just taking things from biology and then being able to do it in a tube to our specifications. So ELISAs are basically using antibody and antigen interactions, very similar to agglutination assays, that happen in the body, in the immune system. The immune system makes antibodies that recognize antigens and then attack, whereas in a lab assay, we take those same antibodies and antigens and we use them to detect or diagnose. So the two types of ELISAs, there's the indirect ELISA and the direct ELISA. So the indirect ELISA is looking for patient antibodies using antigen from a known pathogen. So a very common ELISA that's done for screening for a pathogen is HIV and HIV testing. So ELISAs are very sensitive and very specific and very good so a rapid HIV test might be followed up by an ELISA test. So in an indirect ELISA, you take antigen, in this case HIV antigen, and you fuse it, you stick it to the wells in a 96 well plate that looks like this. So it's just these tiny little, like basically tubes in a plate, and you coat them all with HIV antigen, right? Then you take patient serum and you add it to the well. You can test 96 patients on one plate, so it's pretty cool. You can do pretty high throughput testing. Or you can do multiple tests of the same person so that you get it in replicates and make sure that if you get a positive, that it's truly a positive. So you take serum, patient serum, and you add it to the wells. And then, so let's say this is two different patients. And then you wash out the serum. And if the patient has antibodies towards HIV, it'll stick to the antigen. And if it doesn't have antibodies to the HIV, it won't stick, it'll get washed away. Well, you can't see these antigen-antibody interactions in the well, so you have to add a third ingredient. The third step is to add some kind of like color indicator. So these antibodies that you added to the serum from the patient, so the third thing that you add is an antibody that recognizes antibodies. It recognizes the Fc portion, the stem portion of the antibody. So if there are any antibodies in this well, then that secondary antibody will recognize it. And that secondary antibody has this little enzyme that's attached to it. And when you add a sort of color, a certain substrate, it causes the substrate to change color. It cleaves that substrate and it causes a color change. So positive wells will show this color change, negative wells will not, because there won't be any antibody in there for the secondary antibody to bind to, and therefore there will be no enzyme to cleave the substrate and it won't change colors. So in an indirect ELISA, you are using known antigen to sort of detect patient antibodies. You're looking to see, does the patient make antibodies to HIV or whatever pathogen you're looking for. All right. In a direct antibody, you're actually looking for antigen. So they can do HIV testing this way as well. In the early stages of HIV infection, you can detect antigen but you can't detect antibodies. Whereas later in HIV infection, you can't detect antigen, but you can detect antibodies. So sometimes both tests will be done depending on when the person was exposed. If they think they were exposed recently, they may have a direct ELISA performed, but if they were oh, I just found out my sexual partner from a year ago is HIV positive, then you would have an indirect ELISA. All right, so the direct ELISA, instead of fusing antigen to the wells, you fuse an antibody. You fuse multiple antibodies to the well, and then you add the patient's sample, maybe a blood sample, and if there is antigen there, if they're positive for that microbe, the microbe will stick to the antibody. You add a secondary antibody that has some kind of an enzyme on it, and then you do a wash step, and if there's no antigen there, if the person is negative for that microbe, then that secondary antibody will just wash away and you will get a negative reaction. But if there is antigen there, then the secondary antibody will stick, and when you add the substrate, it'll cleave it and cause a colorful reaction. So what it looks like in a plate, and you often do this in lab, in human biology, anatomy, physiology, microbiology, a lot of our classes do a simulated ELISA where we just use different solutions that change color. It's not a real ELISA, but you can at least see the color change in the wells and identify positive and negative cases. All right, so again, the cool thing about this is we just took a natural feature of the immune system, antibodies recognizing antigen, and we utilized it in a way that we can get meaning and get diagnosis from it. So it's very similar. ELISAs are kind of similar to these lateral flow immunoassays, which also use antibodies, but these are kits that are already sort of pre-made with antibodies to specific antigens, whereas with ELISA, you can set it up with any set of antibodies from a patient. And it's also kind of similar to agglutination assays in that it's also an assay that's using antibody and antigen binding. In this case, it's clumping, and that's giving us a positive diagnosis. So the sort of overall theme of serum testing or immunologic testing is you are using those antigen-antibody interactions to detect a pathogen. All right, and then just a quick video animation of
the enzyme-linked immunosorbent assay, or ELISA, is a commonly used format for serologic testing. The purpose of this animation is to explain how this test works. ELISA serologies are usually done in multi-well microtiter plates so that dilutions of serum can be easily prepared and tested. To better understand how this assay is done, let's take a closer look at what happens in one of the wells of this assay plate. To perform the assay, the wells of the plate are coated with the antigen of interest. For commercial tests, this would be done by the manufacturer of the assay. To begin testing, the wells are filled with dilutions of the patient's serum. If antibodies against the antigen are present, they will bind to the antigen fixed to the bottom of the wells. But only antigen-specific antibodies will bind to the wells. The wells are then washed out to remove all unbound antibodies. Next, a solution of an animal antibody against human antibodies is added. This second antibody is covalently conjugated to an enzyme. The wells are washed again, this time to remove any unbound enzyme-conjugated antibody. Finally, a solution of a colorogenic enzyme substrate is added. The interaction of the substrate with the enzyme on the second antibody generates visible color. The development of color in the wells with a specific antibody can be seen with the naked eye or quantified with an electronic plate reader. The reaction is less intense as the serum is diluted and the amount of antibody captured in the wells decreases. The titer is the highest dilution with definite color development.
So I guess I should also say that ELISAs are a method for measuring titers as well. And I believe that, okay, a couple more things. In vivo testing is really only done, I think the only time I can think of this is the tuberculin test, where they actually inject a substance into your skin and look for a reaction. So that is a way to diagnose for tuberculosis. It's sort of a preliminary diagnosis. There are other reasons why people can have a positive tuberculin test. If they've had many tuberculin tests, it can actually sensitize them to the PPD protein. So follow-up to that would be a chest x-ray. And then some other methodologies that are using genetics are sequencing and microarrays. So microarrays, they use these tiny little chips, so basically like the size of a glass microscope slide, and then using needles can put these tiny little dots of DNA. It's basically like hybridization, but on a microscopic scale, and can do 300 tests at once using one of these tiny little chips, these microchips. And sequencing, DNA sequencing is becoming cheaper and cheaper. So specifically for outbreaks, maybe not for an individual patient, we might not use sequencing unless it was like a very rare disease, but certainly for outbreaks, they might do sequencing. Like for currently with the pandemic, COVID, they are sequencing a lot of the positively identified coronaviruses, and because it's an RNA virus, it mutates very rapidly. So they're able to sort of map out the genealogic tree of these different mutations, these different mutants. And sort of different countries, it turns out, have different versions, different mutants. So that is something that's definitely being done for this new pandemic, to get information on an epidemiological scale of the sequence of these different viruses. Other techniques that can be done, imaging, I said like a follow-up chest x-ray will be done to diagnose tuberculosis. Other types of scans can be done to look for pockets of infection. So they don't necessarily diagnose the pathogen, but they can diagnose the site of the infection or identify infection in addition to like blood tests. Mass spectrometry is a chemistry technique that you can basically use, that chemists can read this output and use it to determine what chemical or protein is present. It's used in forensics to identify chemicals like in soil samples or on clothing samples, but it can also be used in microbiology to determine the identity of a protein from a certain microbe. It's not used very frequently in terms of diagnostics. It's used more in terms of research. And that is the end of chapter 15, all about different laboratory techniques and methodologies.